This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 362, recorded on November 4th, 2015. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How are you doing there?
Hey, it's a beautiful day again. We had a lovely day yesterday. 66 degrees right now, supposed to get up to 73, which is 63 is 17 Celsius. Humidity 63%. Blue sky, not a cloud in the sky. It's great.
Yeah, we also have it's been warm here all week. Warm meaning, high 60s.
Yeah.
Right now it is 19 Celsius, sunny, no clouds, no rain in sight. It's really beautiful.
Yeah.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How are you today?
Doing okay. It's a little unconventional time for TWiV, so I'm still kind of reeling from that, I think.
Your feng shui is all messed up.
Yes. I just had lunch and now we're doing TWiV. Early lunch because I've got a phone interview right after this and just booked. So
I appreciate your flexibility.
It's quite all right.
Also joining us from North Central Florida, Rich Condit.
Hi, everybody. How you doing?
All right. Is it 85 degrees there?
It is exactly 85 degrees here, which is 29 Celsius, and it's kind of humid. And frankly, I'm sick of this. I'm done with 85 degrees and humid. I think it's supposed to go even higher. And it looks like we got another week of this or something. I mean, this is nuts. It's November, right?
Not supposed to, so it's not even supposed to be this in Florida.
This is well, I've seen this before. It's probably not that unusual, but it feels unseasonable. This is like April in snow country, right? When you're kind of done with it,
right.
You're ready for something different, okay? But, 85 and sunny, I guess I can't complain.
Come up to New England.
I can, I will.
It's beautiful weather here, and it's just about peak foliage.
Actually, I was in Washington, D.C. When was that? A couple of weeks ago. It was beautiful. It was really nice.
Yeah, we have great foliage here. I was also in Washington, as was Kathy last week. I don't remember. I didn't see any trees. I guess on that street, I don't remember what the trees looked like. I did, however, run into Bill Nye in the elevator in my hotel.
Oh.
Is that right?
Yeah. I got in, and it was full. There were a lot of people in it, and he said, "Come on in, you can fit," in that aggressive voice that he has. I got in, and I'm looking at him, and then I got out, and there was the lady from ASM who publishes our books. I said, "Come here," and she said, "Yeah, I think so." I didn't even have to say anything. And he was running.
Was he wearing a bow tie?
No, he just had a white shirt on without a tie.
Well, that's why you didn't recognize him.
He was incognito.
And he had his assistant, and they were running around. Well, she was sitting down. He was running around. I wanted to do a selfie and give him a TWiV card, but he seemed busy. Too
bad the elevator was full because you couldn't have had him do the click either.
Yeah, I didn't respond well, but it was cool. I'm like starstruck. It's weird, right? I say, "Look, it's Bill Nye," and I told people, and people are like, "Come on!"
It's cool.
I think it's really cool. On my hotel, right?
Yeah.
And we're doing this at a weird time. It's Wednesday, 11 a.m. because I have to move my wife back from North Carolina on Thursday and Friday. Her company was sold. It's in the news. The name of her company is Cardioxel. So that's the end of the North Carolina adventure. Although I will be going down there next semester to give a talk. I think Blossom Domania is going to invite me, and my wife goes, couldn't you have done that while I was there?" No, it's not my choosing. That's the way it goes. So we're all a little discombobulated because 11 a.m. It's Wednesday. Friday's best day to do TWiV at 2 p.m.
Yes.
Well, we've gotten used to it also, right?
Right.
People get used to things,
Yep.
Like, tell me about it.
We're thinking about selling our house, and this is really throwing me off because I lived there since 1989.
Wow.
And
why are you thinking about selling your house?
Kids are finishing school.
So the kids will all be in school. It's a big house. The real estate taxes are really high. It's not clear we can afford it. And
you could shorten your commute.
I could. So I want my wife to work somewhere up in Westchester, maybe, and we could move up there. And it would be a lot shorter. So we'll see. But it's really messing with my head because we lived there a long time. We renovated it exactly the way we want it. It's gorgeous.
Yeah.
There's a fire alarm.
Oh.
I don't know if you guys can hear that. Can you?
I can hear a little bit. A voice in the background. Yeah.
I don't even know what they're saying. Okay, I'm going to just stay in my office until they throw me out. This is great. This episode, by the way, is sponsored by ASM Education, the ASM Education faculty programs, and the ASM Leaders Inspiring Networks and Knowledge program, that's called LINK, is offering a 4-part webinar series for educators and researchers. This series is called ASM MOOCs, ASM's version of MOOCs, massively open online courses. And the theme is successful integration of research and teaching. There's one left in this series, and it is called Publishing Ethics and Responsible Conduct of Research with Roseanne Sandry Golden, University of California, Irvine. November 18th is also the editor-in-chief of the Journal of Virology. Now, I'm not going to give you a link because I want you to go to the show notes and use the link that they gave us so that they know that the traffic is coming from TWiV. But you have to register for this, and it's coming up November 18th. So if you want to do this, and it could be cool. It's a webinar. You don't have to go anywhere. You can do it in the comfort of your home. Yeah. So check that out. And we have some follow-up. Kathy Spindler has some follow-up.
Oh, I don't have the... Oh, there it is.
You don't have it in front of you?
I didn't have the Google Doc open, but... yes, I listened again to for the first time because I wasn't on, to the TWiV that you guys did last week about the rat virus and so forth. And I think it's the second time that Dickson claimed that Harry Ginsberg played football at Michigan. And I fact-checked him on it before. Harry Ginsberg, according to Wikipedia, got an undergraduate degree from Duke University. Mm-hmm. And I also checked a list on Wikipedia of Michigan football players, and Harry Ginsberg is not there. So,
did he play football at Duke then?
That I don't know.
I presume so, because that's what he did play football.
Yes, I think he did, and I thought I recalled that he was a Southerner, and
he had an accent.
So
sure.
when I heard Michigan, I thought, wait, yeah, really? So
the thing is, Kathy, we correct Dickson, but he just repeats it.
Yeah.
Oh well.
Yeah, but you got him right on which Norwalk named Norwalk.
Yes, he's finally straightened that out.
Yeah.
Yeah. So, and then the only other thing was that there was a little confusion about when our department was formed, and it was formed in 1902, not 1880 something.
I went back because I said 1888 or something, and I got that from the New York Times article that Vincent said, and I sent, and I looked back at that, and it said he helped establish bacteriology or a bacteriology lab at Michigan in 1888. So that was not I mistook that for the beginning of the department.
Right.
Did you like our presentation of the rat virus, Kathy?
It was great. I think the story is really cool. I told people about it at study section. I mean, just grinding up the tubes with the labels and everything.
It's so weird.
It's awesome.
It's a great story. Yeah. It's cool. And then Powell, who wrote that article, he's just terrific. We should possibly have him on at some point just to chat about history of microbiology. Mm-hmm. All right, we have a few other follow-ups. Walter writes, Professor Racaniello, we have been hired to find the lost banner you photographed today. What cross street is your office? I assume you are at Columbia. Thank you for your help already. And Walter is a captain in the Seatow Central Hudson River Company. A captain! So this came in Friday evening, and it refers to that banner that I photographed during TWiV. And Banner put it on Twitter, and I guess they were looking for it.
They were looking for it.
It's pretty cool. I wrote to him, and I gave him an uncropped picture so he could see the shoreline, because it was a little south of where I am. And he asked, did it sink or did it float? And I said, yes, it seemed to sink immediately. And he said he didn't find any other pictures except mine online. And they came up on Saturday or Sunday, and they couldn't find it. But they were going to go back. I think it must be costing money for them to salvage this thing, right? I don't know how much it's worth for a banner.
Yeah.
I don't know. There's the issue of how much the banner is worth, and there's the issue of how much they might have to pay if it gets counted by the Coast Guard as polluting the water.
Yeah.
I don't think there's anything in a banner that's going to cause a big problem. It's not like oil, but it's I don't know.
Maybe they don't want it on the bottom of the river there. I don't know how deep it is. Does anyone have any idea how deep the Hudson is?
It's pretty darn deep. It's well, if you look at the shipping traffic going by, I don't know how deep it is right where the banner sank, but parts of the river are 50, 60 feet deep.
Yeah.
I mean, it's diveable depth if you pick your tide right and you have the right visibility. Probably the thing to do would be for Cito to just go and drag a grapnel across the bottom.
Right.
Because the banner, I mean, it's a big object that would snag easily. Of course, there are about a bazillion other big objects that would snag easily on the bottom of that. Too, so.
I thought that was cool.
Yeah, I can't imagine what you would come up with if you grappled the bottom of the Hudson.
Yes.
Yeah.
I am all too familiar with Cito.
You are?
Yes.
I have been towed by Cito.
Really?
And on one occasion, and escorted by them on another occasion. And I can tell you that they're not cheap.
They're not cheap.
So what you do is you buy insurance.
Yeah.
But I doubt if a guy flying a banner has insurance with Cito.
That's right. I did see them on the bay this summer while we were sailing. Yeah, and I thought that they might be expensive.
Well, the banner company would certainly have insurance. It wouldn't necessarily cover that, but it'll probably cover something.
Anyway, good luck.
Yes, good luck, Walter.
You haven't heard back from Walter, huh?
I heard back Monday, and he said, they hadn't found it yet.
Okay.
I don't know if he'll bother to tell me if he does, but anyway, I thought it was a cool thing.
Yes.
Mm-hmm.
Alan, can you take the next one?
Sure. Judy writes, too late for Halloween, but 3 vampires walk into a bar. The first 2 ask for blood, the third asks for serum. Bartender says, let me get this straight. 2 bloods and a blood light.
There you go. He groaned.
Yeah.
Judy's in San Diego, where it's, at the time of writing, 81 Fahrenheit, 27 Celsius, and sunny, no wind. And this is the last day of October.
81. Now, when I was there, it was 68.
Yeah, that's warm for San Diego.
Couldn't you have at least been 72?
Wow. Yeah.
The last one, Rich Condit.
David writes, follow up to episode 361 where a couple of incorrect paleontological statements were made. There are no known marine dinosaurs. Mosasaurs are reptiles, but not dinosaurs, as are other marine reptiles such as
plesiosaurs, ichthyosaurs.
You must be a dino freak. Plesiosaurs, ichthyosaurs, and turtles. Also mentioned were pterosaurs. They are also non-dino reptiles. There have been some semi-aquatic dinosaurs, but none that have lived predominantly in the ocean. However, there have been rare cases of land-based dinosaurs found in marine deposits. Theory is that they were entangled in plant mats that floated out to sea during a storm. Wow, that's a stretch. The bones then fell to the bottom of the ocean after the meat of the dinosaur and the mat decays away. I don't know if that is the case with the Japanese dinosaurs. One of the topics discussed on several TWiV episodes combines 2 of my interests, paleovirology. Please continue including such discussions in future episodes along with viral evolution. From my online research, the oldest viruses from endogenous retroviruses in present-day insects is about 300 million years ago, about the time reptiles began, but before dinosaurs. Maybe one day we will be able to answer the question, did T-Rex get the flu? It is a nice 75 degrees Fahrenheit, 25 C in Austin. That's from David.
Well, many years ago, before we had this dating by endogenous viruses, the sequence of herpes virus genomes, the clock that was established from that, put the origin around the time of when the dinosaurs were around. So dinosaurs probably had viral infections, although, of course, we can't prove it. But they were around at that time. I think somewhere online there is a picture of T-Rex with lesions on its face, reflecting that.
So when we're talking about origins, we're talking about the origins of viruses?
Not the origins, but they were there. I mean, the herpes viruses were there, but origins probably way before that.
I'm on board with Eugene. I got in my head, viruses are in the primordial soup.
Yes.
Yeah. And now that the ICTV has said that a viroid is a virus, there you go. Naked nucleic acid, right?
Yep.
Mm-hmm.
That's what Jens said anyway. All right, we have a snippet suggested by two people. Neva from Buda writes, VIRCAP seek vert, really? Hi, TWiV gang. Wow. The idea that the methods in this article can identify all and every virus in a sample is really terrific. Really? What a leap forward in diagnostics and discovery. Can something like this work for microbes or parasites in all their diversities? Sounds very cool. I bet you have gotten many notes about this and hope you can explain a bit with your usual verve. Neva in Buda, where it is 91F and partly cloudy at 6:30 this evening. Not 6:30? It's bloody 91?
Wow.
Yeah, that's we're talking Texas, dude.
Huh.
That's what happens in Texas. And that's pretty recent.
Well, Neva, you don't need to do this for microbes or parasites because they have ribosomal RNAs that you can sequence very easily. And this is the problem why it's not easy to find viruses, because they don't have any conserved sequence like that. Ribosomal sequences are conserved in every organism that has them. So you can find microbes, you can find parasites, but this is why this is cool for viruses. So we'll talk about it. And Marcia writes, Hi there, TWiV people. I came across this article today since it's about testing for viruses. I immediately thought of TWiV. I would love to know more about how this works and maybe how it was developed. Regards, Marcia in Salem, Massachusetts. I think we've heard from Marcia before. That's far from you, right, Alan? That's on the coast, correct?
Salem, yes, that's out on the coast.
That's where the witches used to be, right?
Witch hunt. Yes. And in fact, still are if you believe some of the locals.
That's where the witches were persecuted, right?
Yes.
It would put you in water, and if you drowned, it proved you weren't a witch.
That's where a couple of dozen people were hanged because a child had a temper tantrum, basically. Yes.
And the story by Nathaniel Hawthorne, the Scarlet Letter, took place in Salem, right? Does anybody know that? Yeah,
so we can fact check you.
Guys, didn't you read the Scarlet Letter in high school?
Yeah, many years ago. Yeah,
it is a fiction, a romance in an 1850 setting. Where is it? Hester Prynne, Arthur Dimmesdale.
Oh yeah.
We read this in high school. My mother was a big fan of the Scarlet Letter. Let's see where it took place. Salem, Massachusetts. The nameless narrator was the surveyor of the Custom House in Salem, Massachusetts. Oh, and you can go and see the house that it's based on, in fact. Okay, I know, I'm wandering off. My apologies. It just goes to show that scientists have other interests as well. All right, the paper in question was published in MBio. It is called Virome Capture Sequencing. That's what VIRCAP-seq stands for, and VERT means vertebrates because they're looking for vertebrate viruses. VIRCAP-seq VERT. Don't let scientists make acronyms.
No, or name things in general. That's so hazardous.
Enables sensitive viral diagnosis and comprehensive virome analysis. The authors are Breeze, Kapoor, Mishra, Jain, Kumar, Jabato, and Lipkin. They're
and
here.
Because this is in MBio, anybody can download it.
It's open access.
Yep.
And this is done here at the School of Public Health at Columbia University. Now, we've had some of these people on TWiV. We've had Thomas, we've had Amit, and we have had Ian Lipkin, of course.
Of course. All
right, so the thing here is deep, high-throughput sequencing is great for discovering viruses. If you take some water or soil or mountain rock or whatever, you can sequence it and discover viruses. But it's not good for diagnostic purposes.
Mountain rock? Where did that come from?
I don't know. I just pulled it. I was trying to think of diverse. I know there are bacteria in rocks.
Okay.
So if there are bacteria, there must be viruses, right?
Right.
Sure.
And those bacteria, by the way, are thought to divide every few hundred years.
Wow.
How would we know this? That's a good question.
In fact, there's a phenomenon out in the Southwest. You can see it on the rocks called desert varnish.
Yeah, right.
Really, really gorgeous streaking of the rocks that occurs, and it's because of the action of bacteria in and on the rocks over periods of millions of years.
Yep. All right, so back to the paper. You have a diagnostic laboratory. They get samples from people who are sick, and you want to know what's in the samples, right? And if it's a virus infection, you don't use high-throughput sequencing typically because it's expensive, it's complicated, it's not very sensitive just to take the whole sample and try and sequence and find a virus. I mean, people are working on this, and certainly very fancy labs have high-throughput sequencing capability. I did a TWiV out in San Diego with a lady who heads the clinical lab at UCLA Medical Center and a guy from CDC who does high-throughput sequencing. And she said they have one in her laboratory, and they're working on developing this. So this paper, they try and address this. Can we make a method that allows us to use high-throughput sequencing to take a sample from someone who's ill and say you've done a bunch of PCR for directed discovery, and it comes up negative. So right now you say, okay, we think this person has a herpes infection. You can use primers to do PCR for herpes viruses, but what if it's negative? Then what do you do?
You have to know what you're looking for in order to find it is the problem.
Yeah.
Right.
So
I think there are also a number of clinical diagnostic kits where they will do PCR for a certain number of the most common infections that you might find. Okay, so you can do it in a semi-selective fashion.
Right. You can have a collection of respiratory viruses, for example.
Right.
Yeah.
Exactly. Yeah.
Yeah. In fact, CDC has all these probe collections, and they're directed against lots of viruses. An interesting example here is that last summer there was this outbreak of enterovirus 68 in the U.S., and the probe for that wasn't in the CDC collection, and so all these laboratories were coming up negative with their, respiratory virus probes. So they did some deep sequencing and discovered this virus, and I think the CDC did that. And then the CDC included the proper probe in their collection, and then people started finding it all over the country.
Right.
So in this paper, they designed a strategy to do this. They designed it to discover known viruses that can infect vertebrates, including humans. And here is the amazing part of this. They designed oligonucleotides to represent all viral taxa with at least one virus known to infect vertebrates, all right? And they ended up making 1,993,176 oligonucleotides.
Call it 2 million.
You have to raise your pinky slowly toward the side of your mouth and say 2 million oligonucleotides.
Now, here's the thing. When I order an oligo, say a 20 mer, it costs $3. Let's say the oligo is $1. He gets a discount. It's $2 million. It's still he can't have paid two. He didn't pay two.
No, I'm sure they either made their own oligos or they did some kind of a volume discount with somebody.
Yeah.
So they have oligonucleotides, many and they do the whole genome pretty much up and down. They have a few pictures here where you can see that the whole genome is pretty well represented for a couple of the viruses they selected. But
as I understand it, it's not like they picked out. The way they did this was to pull out of a database, I guess the EMBL database, viral coding sequences,
right?
Just sort of at random, and make oligonucleotides for those. They didn't say we want oligonucleotides to cut through this virus or this virus, or they just took all the viral sequences and made oligonucleotides. And after that, they went back and looked at, okay, this virus, what oligonucleotides did we pull out of this that cover this virus, and what sort of coverage do we have?
Exactly.
And when they look at that, they see, oh, well, it's pretty good coverage.
Yep.
So this confused me just a little bit, and something Rich just said, which it reads right out of the paper. Coding sequences were extracted.
Yes.
So does that explain the part of figure 1A where they don't get the 5' untranslated region
Yes.
covered?
I assume so, yeah.
Yes.
Okay.
Yeah, it's only coding sequences.
Right. And they specifically state that they did not get the like the 5' untranslated region and stuff, and that is why.
Right. Okay. And then one other question then, and this includes any and all sequences, whether a virus has been isolated or not, but just something that's been identified as a viral sequence. Is that correct?
I'm not entirely sure about that because they didn't explain sort of how they mined the database in detail in the paper, and I didn't look at the supplementary information, so I'm not sure. Do you know?
They basically say they want to represent all taxa containing at least one virus known to infect vertebrates. So you can't just have a sequence and not know what it infects.
Right. You can't just dump in the whole discovery.
It has to be something that's
database.
known to infect the vertebrate. So that's pretty stringent, right? And they excluded plant viruses and insect viruses from this as well.
And the other thing is that there's either for sequencing errors or variability in the virus sequences out there, for a given region of a given virus, there's heterogeneity in sequence among the oligonucleotides.
Right.
And that's not necessarily a bad thing, okay, because it gives you a broader capture.
So they say to address sequence variation, oligonucleotide sequences were retained if they diverged by more than 90%. So among related viruses, I suppose. But as we'll see, this can handle pretty good variability. It can't handle total variability, of course, but up to a certain percentage, which we'll see later. Now, my understanding is how this works wasn't entirely clear, but tell me if I got this right. The oligonucleotides are made with biotinylated triphosphates? Correct. Okay.
Or they got biotin on them somewhere.
They got biotin on them, and therefore what they do is they take a sample, they amplify the nucleic acid by PCR, random prime PCR, so they can have more DNA. And then they denature it, and they add the oligonucleotide, which will then hybridize to the sequences in the sample. And then they can remove any nucleic acid that's hybridized to their oligonucleotide using streptavidin, which binds with high affinity to biotin. And the streptavidin is connected to a little bead that can then be pulled out with a magnet.
Right.
Right.
All correct. The only thing I would add is that in the preparation of the sequences from their sample, it's the preparation is as you would prepare something for sequencing,
right?
Which involves adding linkers and actually making a library so that the sequences that you're pulling out are ready to be sequenced. Because I was concerned about how you would distinguish between sequencing the clinical material and sequencing the probe that you've made to try and isolate the clinical material.
Right.
And the distinction there is because since you've added linkers to the clinical material and the sequencing is based on priming off of those linkers, you can distinguish between the clinical material and the probe that you use to pull it out.
Right. So they do a series of basically testing this system to see how it works. They usually will use their VIRCAP-seq, which is it's basically to enrich any viral sequences that are present versus a standard way that you would prepare the material, as Rich said, for high throughput sequencing.
And I want to make one other point, and that is that this notion of enriching for sequences by hybridization to a biotinylated probe, that's not novel.
Right.
Okay. This is, as a matter of fact, you can buy kits now that do this. Okay. The novelty here is this incredible
2 million
2 million oligonucleotides that represent sort of the whole virome, the global virome.
Yeah. That's the novelty, and it takes a little bit of chutzpah to do a 2 million experiment, for sure.
Oh, yeah.
Absolutely.
Yeah. And also the vision of applying this to a clinical sample in a diagnostic sense, that's, of course, novel as well.
I guess in the end, you're going to sell a kit which has these 2 million oligos in it with instructions, and in a clinical lab, they could readily do it. Right? But it has to be easy. It has to be simple.
Someday it'll be a robot. You take some serum and you stick it into this robot, and it gives you the sequences at the other end.
It has to be easy, highly reproducible, and well documented.
Yeah.
And cheap
to use clinically. Yeah. And cheap, yes.
Yeah.
So in one example, they took extracts of human lung tissue or whole blood, and they spiked this with different amounts of nucleic acids with virus sequences in them, large and small plus and minus strand, segmented, non-segmented DNA and RNA viruses. And then they process them according to either just high-throughput sequencing or their VIRCAP-seq.
Right.
All right. And they say VIRCAP-seq gives a 100 to 1,000-fold increase in viral reads and a reduction of background reads from 99 to 68%.
So they're cranking up the signal without the noise.
Yeah, because you get a lot of host signal when you sequence, and you don't want any of that. You have to throw it away, and typically you throw it computationally, subtract it. But if you can get rid of a lot of that to begin with, it really helps. So it goes from 99% to 68% in the lung and from 99% to 38% in the blood, which I guess makes sense because there's less complexity in the blood, I suppose. Mm-hmm. Average coverage also increased with nearly full-length sequences over 95% obtained for all viruses that they put in there. That's pretty good.
Yeah.
And then they want to know how much nucleic acid could you detect by this. And they got over 90% genome recovery for the viruses with 100 viral copies in 50 nanograms of blood nucleic acid or 1,000 viral copies in 100 nanograms of lung nucleic acid. So 100 copies is very good. You're not going to detect 1. You're not going to detect 10. That's tough. But if something is causing disease, it should be there at 100 copies, I would guess.
Yeah.
Okay. What else have we got here?
And that was whole blood, by the way, not blood light.
Blood.
Yeah.
Very good. Very good. Okay. Then they tried it with fecal material, which they say is challenging for viromic analysis. I like that.
Yes.
It's frequently challenging. They've tested a sample of fecal pellets from bats known to contain rotaviruses. And they got they tried 4 different ways of looking at this, and their VIRCAP-seq gave them the highest read count.
And I assume they did that because if you're in Ian Lipkin's lab, you just happen to have bat poop in the freezer.
Do.
Oh, yeah.
You do.
They got all kinds of stuff.
In fact, they were able to more precisely identify this rotavirus than had been previously possible. They didn't have enough sequence before, and they were able to do that. Okay, how about novel sequences? To test the capacity to find novel sequences, they took a liver homogenate from a deer mouse that had been infected with a rodent hepacivirus. And the genome has a less than 65% identity with the sequences used to design the probes. Okay, less than 65% identity with the VIRCAP-seq probes, but they pulled it out.
That's important to do because you want this to be as wide a net as possible.
Exactly. So less than 65 is pretty good. But again, it's not going to pick up something that you don't have in the database, right?
Right.
It will not
This will probably not find an entirely new virus of a whole new, type that's never been seen before.
Yeah.
But if it's close, that 65%, I mean, that's not terribly closely related. So if it is a known, a known genus at least, you're going to probably find it.
Right. They also asked whether you could do sample multiplexing. All right.
So that means, if I understand this correctly, that means taking several different samples and making a library with each one. And in this case, you do what's called barcoding them, which means that when you add the linkers in the library preparation, there is a unique sequence in the linker for each of the different samples, so that when you actually do the sequencing, you can do them all in one tube, but you can distinguish which sequence came from which sample. Have I got that right?
That's right.
Yes.
Right.
Okay. So this means this takes the expensive part of the test and allows you to pool multiple samples into that part of the test.
Right. Which you can do with high throughput sequencing also.
Right.
Because you barcode
But then the question becomes, can you really do this? Because you got all these sequences. There might be competition if you among sequences from different samples and that kind of stuff. And does that mess it up? And I guess the answer is pretty much no. They can do the multiplex.
They can do it, but they say that when you have 7 together, it was better than 21. You get better genome coverage when you only have 7 as opposed to 21. So,
so some of that competition is a factor.
Yeah,
right. So there's a limit.
Right. So, but if you multiplex this thing 5-fold, you just save 5-fold on the price of the sequencing part.
And then to test this out, they had a set of 23 serum samples collected from multiply transfused hemophilia patients. These people get lots of transfusions, right? So they're often positive for many viruses, and these are known to contain hepatitis C virus, GB virus C, human HIV, and torquetanovirus, TTV. And they barcoded them and put them together. 9 in one pool. They had 9 samples, and then another one they mixed those 9 with 14. And in both the 9 and the 23-plex pool, they got all the viruses out. They identified them all. So that's basically it. It's a new method for discovering viruses in potentially in clinical samples. They say it costs $40 per sample.
In a 20-plex sample, yeah.
Now remember the paper we did where they look for antibodies in people to discover what's infected you? They said that was $10 per sample, right?
Mm-hmm.
Right.
I forgot what they had to do something also involving lots. Peptides they had to make for that. Right. Remember?
Right.
And the antigens. So it's
this is much better because just looking for antigens, you could get cross-reactivity. There's a lot of noise you could get in that. Whereas if you're looking for genetic material, you're really closer to seeing, this person really has a virus.
Right.
In that other paper, were they looking for antigens in the clinical samples? Antibodies. So in that case, you could see everything that a person had mounted an immune response to at any time. It doesn't tell you what the active infection is.
That's right.
Yes, right. So the advantage of the antibody test is it gives you sort of a history. The disadvantage of the antibody test is that it's a cloudy history because you can have cross-reactive antibodies that would be, this could react with Ebola virus, but it could also be because you were exposed to some weird protein.
Right. And if we remember from that paper, they had underrepresentation of some viruses that they expected to be highly represented, right?
Yes.
So it's not perfect. So probably nucleic acid would be better.
Right.
But this, of course,
it depends on the type of research question you want to ask.
Yeah. Well,
I think in a clinical setting, this would probably be more useful.
Right.
Yes.
This is a good one. So this obviously has to be further developed, but because they say in the discussion, this is a promising tool for diagnostic and research application. Sensitivity similar to that with targeted real-time PCR. Okay.
One of the things that interested me about this technically is that they did a bunch of comparisons of different ways to prepare the sample, because one of the things they point out is that in current techniques, there's a lot of stuff that people go through to try and enrich for viral sequences, like filtering and treating with nucleases to get rid of unencapsulated genomes and that kind of stuff. And they do some of that with these samples before they make their libraries and do their capture. And in general, what they find is that they're better off not doing anything. Okay,
Yep.
Which interests me. Probably all that processing in this particular context actually compromises the sample. You're better just to take it raw.
Right. And also to point out, besides the sensitivity comparison with PCR, that it can handle for up to 40% nucleotide divergence, as we saw in that one example. So that's pretty good. All right, so there you go. I hope we did it with our usual verve, Neva.
Yeah.
Was that vervacious?
Is that a word?
Sure she'll let us know.
I just made it up, but
Okay.
if enough people use it, we could make it a word, right?
Right.
Let's look it up. I bet.
Are you going to interrogate Wikipedia?
I'm going to be vivacious and look it up. It's a website about food, It's a restaurant in Portland. Wow, it's a food thing. Cool. But it is not having to do with verve in the sense that I meant. All right, we have a paper today that came across my digital desk a while ago when we were talking about changing the codon usage or the dinucleotide frequency or whatever you want to call it in viruses to make them attenuated. And I waited for it to be published. And I think it's really interesting. It's called Importance of Codon Usage for the Temporal Regulation of Viral Gene Expression. And it is from the laboratory of Ron Desrosiers, and Ron was the advisor of Blossom Domania, if you remember. He was when he was at the New England Primate Laboratory. He's now moved to the University of Miami. So he has improved his football score. Which went from 0 to more. And the authors are Young Shin, George Bischoff, William Lauer, and Ron Desrosiers. Edited by Stephen Goff.
Is that how you discovered this paper?
Yeah, he sent it to me a long time ago, and he said, I didn't send this to you. And then he wrote Desrosiers, and Desrosiers said, yeah, they can talk about it whenever they want. I just waited for it to be published. Now,
it is also open access, by the way.
Very good. Open access we like, so people can read them. Now here we're looking at the glycoprotein genes of two different viruses.
Two very different
viruses.
Right. One is a rhesus radenovirus, which is, if you remember from what Blossom told us, I think this was isolated by Desrosiers, and it's like the ortholog of Kaposi's sarcoma viruses of people. So you can infect rhesus macaques with this virus, RRV, rhesus radenovirus, and you have an animal model for infection. But it's very much like Kaposi's sarcoma, and then SIV, simian immunodeficiency virus. And what's been noted before this paper was that the genes encoding the glycoproteins, two of the glycoproteins, well, for SIV, the main one, GP160, and for RRV, I suppose like most herpes viruses, it has many, and this one is called GH. These are made late in infection, which makes sense because that's when you would have enough material to make a new virus, right? So they're regulated, they're temporally regulated. But what has been previously found is that the codon usage is different from these, from normal codon usage in, say, a cell, right?
Right.
These two proteins have a different codon usage. What they say is, and this previously had been found to influence production of the protein. So they wanted to explore this here because they have this idea that maybe this weird codon usage is important for the regulation of the production of mRNAs encoding these proteins. So they do a bunch of experiments where they have a gene in a plasmid with a promoter. They put that into cells, and then they do Western blots to look at protein production. See how good I am to say production?
Yeah.
I'm with it. I don't use the E word anymore.
You're not very expressive.
No.
Yeah, I know. I'm sorry about that. Anyway, they do an experiment where, so SIV and SIV GP160 and GH have these different codon usages, not only different from the rest of the viral proteins, but different from each other as well. So first they take SIV GP120 and they make it GH-like in its codon usage, right? So they make it like the rhesus radenovirus. And then they make GH like GP160. And they don't have to do a lot to do this. In SIV GP160, they change 6 codons. 6 codons in general. That turns out to be 93 codon, or 10% of the total codons in the coding region.
And if I understand it, that's not all of. They could have done more. Right. There are more codons that are different than that, but they chose the ones that are like the most different, so that they could swap them out.
And these are distributed evenly throughout the coding region of the glycoprotein, right? Now, the other thing I should tell you is that the transcription of these genes is normally regulated by viral transacting proteins,
Right, and these are normally expressed, both these viruses, as do many others, mostly DNA viruses, go through an orderly temporal expression of genes where they have early and intermediate and late gene expression. And these, being structural proteins, are typically late genes.
Correct.
And are subject to, they're turned on by earlier virus protein products that are transactivators, turn them on somehow.
Right. So for SIV, the transactivator is called REV, R-E-V. And for the RRV, the radenovirus, it's ORF57. Shows you the different philosophies of herpes virologists versus retrovirologists. Mm-hmm. ORF, they call them ORFs. Okay. So what happens? Basically, if you take... This was amazing to me. If you take either one of these glycoproteins and just put the coding sequence behind a promoter, a cytomegalovirus promoter, a strong promoter, and put it into cells, you don't get any protein production without the transactivator present. Isn't that weird?
Yeah, it is weird. It's a standard transactivation assay. It's particularly weird given, as we're going to discover, that it's codon based, because it's hard mechanistically to understand how this is happening. I mean, regular transactivators where it's a transcriptional transactivator, you're right. We don't have any special upstream sequences. It's just a garden variety strong promoter. So yes, it's weird that you see the specificity. It's also, to me, noteworthy that there's no other viral proteins here. You're just transactivating. That's
right, just the plasmid.
You're just adding the plasmid with the coding sequence for the protein of interest, the glycoprotein, and the transactivator. Nothing else. There's no infection, no nothing. So the transactivator by itself, or with whatever cellular proteins might also be required, can do this job.
Right. Sorry, go ahead.
No, I was just going to say, when I was thinking about this, I hadn't heard about this. For instance, if you put one of these genes behind the immediate early CMV promoter, you don't get expression. And I just wonder how many graduate students and postdocs, beat their head against a wall because nobody reported that we couldn't get this thing to express.
Yes, exactly. Right. That's why I say this is weird, because hasn't someone tried this and they couldn't get it to work, and you go to the PI. I can't get this to produce protein.
No, of course you can.
Of course you can. Right. You're not. You don't know what you're doing. Do this. Do it again. Do it 20 more times.
Well, it's interesting because a common thing to do. Well, maybe it's common for me to do nowadays when you can't get something to express is to codon optimize it. Okay. This is particularly true for pox viruses because pox virus genes evolved in the cytoplasm. Okay, and so they often contain signals like cryptic splice sites and that kind of stuff that if you try and express them in the nucleus in a transfected cell, get in the way of expression. When you want to express a poxvirus gene outside of the context of a poxvirus, first thing you do is you get somebody to make you the gene with the codons and all the signals optimized for expression in a nucleus.
That may, in fact, have what led them to the previous discovery of this. They probably they probably looked at the codons and said, "Whoa, this is not right."
Yeah, right.
So now you take GP160 with a CMV promoter, the normal GP160, you don't get any protein production. But if you put REV into that system, if you add another plasmid encoding REV, now you get induction. If you have a plasmid with ORF57, it does not induce it. All right. So this is all the controls so far. Now you take
so that indicates there's some specificity.
Right.
The fact that ORF57 doesn't work, but REV does.
Right.
Now you take the codon modified GP160. Now you've made it look like RRV. And when you add REV, you get very little protein production. But when you add ORF57, boom, you get a lot of protein. See the gel? It's right there. It's beautiful. Mm-hmm. And so you can switch the induction pattern, whether it responds to REV or ORF57, by changing the codon usage.
So somehow these transactivators are responsive to the specifics of the codon usage in the gene.
This is far out.
It's impossible for me to think about. Okay?
I would never have predicted this.
And they avoid thinking about it entirely in their discussion. Okay?
Well, at the very end, they kind of throw us a bone. But.
Yeah, they do it a little bit. They do the same experiment basically with the RRVGH coding sequence. They make it resemble GP160, and now that can be induced by REV and not ORF57. So it works both ways, which is nice because the codon usages are different, right? Then this is a cool... they look at RNA to make sure that all these results are at the level of RNA, and they are. So the protein expression correlates with RNA levels. So this is a transcription at the level of transcriptional regulation.
Well, I wouldn't necessarily conclude that.
Maybe it's both. Maybe it's both.
Well, when you're looking at RNA, you're looking at steady state levels of RNA.
Yeah.
And it could be that if an RNA is not translated, it gets degraded.
That's true.
So it could be that they actually get made, but only the ones that are translated persist at a fairly high steady state level. If you really want to know if it's transcriptional, you're going to have to look at active transcription somehow.
Fair enough. Yep. They took a luciferase gene, which is usually used as a reporter, totally different gene from these glycoproteins, and they modify it to look like either GP160 or RRV in the codon usage, and guess what? It responds to the appropriate transactivators. So this can work with other than the glycoprotein genes.
By the way, just as an aside for the Calebs out there who are not doing this every day, luciferase is firefly luciferase. It's the enzyme that, in complex with a certain substrate and a lot of ATP in the tails of fireflies, makes them blink at you. Okay, so you can take this enzyme and appropriate substrate and ATP, and it will make it'll make light in a test tube. That's why it's a good reporter gene. That's how it's been used. I know this because I used to grind up firefly tails.
Really?
Yeah. I mean, I'm an old guy, right? I did luciferase assays as a graduate student, and to do they didn't have cloned. Well, nothing was cloned. Okay, and you didn't even have purified luciferase enzyme. You could buy firefly tails from Sigma. As a matter of fact, the story is, and I think that this is true, that in St. Louis, where Sigma headquarters was, they would pay little kids to go out and catch fireflies. All right, and you could even join the Sigma Firefly Club. We got hats for the people in the lab. That was the Sigma Firefly Club with a little insignia on it and stuff like that. And then they package up these firefly tails, and I would take the firefly tails and grind them up and make a homogenate. And that was my source of luciferase. I think it also had luciferin, the substrate, in it as well. So all you had to do was add ATP, and you'd get flashes of light, which you could actually measure in a scintillation counter. So it was a good assay for ATP.
And if you Google Sigma Firefly Club, the first hit you get is a 1987 United Press wire story talking about Sigma Chemical Company here pays a penny for each firefly it receives.
Go.
Receives them by the thousands. And the Sigma Firefly Scientists Club, and they talk about the people going out and collecting these fireflies.
Next experiment, they want to know what part of these glycoprotein genes are important, because as the codon changes are scattered throughout them. So they divide up the codon optimization, whatever you want to call it, to different parts of the proteins, and in the end, the N-terminal region of GP160 is the most important part, either the 1/3 or 1/4, depending on how much of a signal you want to get. So whatever is conveying this ability to be properly transactivated, it's in the very N-terminus of the coding region.
So I wonder if that's going to be a general property of this. That is, the N-terminus, because I'm thinking translation. I'm not thinking transcription. Because it's a codon-based thing, I'm thinking translation. And I'm wondering if you start to translate something and you get through the sequence and then you run into the codon issue, by then the deed is done, okay? And it's only if it's in the N-terminus where translation starts that you see this sensitivity.
So you're thinking that with the wrong codons you get poor translation, and then that kind of shuts everything down?
Right, and somehow the transactivator stimulates transcription or translation through that particular arrangement of codons.
It could be.
So I'm thinking this N-terminal thing is in fact a clue maybe to the notion that the mechanism is translation. And I'd be looking to see if these transactivators like stuck to ribosomes or something like that.
Mm-hmm. Yeah, I'm sure they are. They also do an experiment where they make a virus. They make rhesus radenovirus with the GP160 gene from SIV, optimized so that they respond to ORF57. And these can infect monkeys. And they say for the first time, we get antibodies to GP160. They've never seen this before when they infect with SIV, I suppose. So they're very excited about this. They say, first time we can get anti-GP160 antibody responses with this vector system. So I guess in terms of vaccination or antigenicity, that's interesting to them.
Absolutely.
And then they say, well, how general is this? Let's look in other virus families. So they look at 6 families containing persisting viruses. And they look at... let's see, I can't see here. Papillomaviruses, SV40, adenovirus 2, and parvovirus B19. All 6 structural gene products of these viruses showed skewing of codon usage. These are persisting viruses. Mm-hmm. So let's look at acute non-persisting viruses. Poliovirus, measles virus, Ebola virus, rabies virus, and yellow fever virus. Guess what? No problem. The codon usage is more aligned with that of the human exome. So they're wondering, is this a general property? Do persisting viruses have these structural proteins that are differently codon optimized? And then they do an experiment. Does this kind of recoding help other viral proteins get produced? So they take VP1 of SV40 and papillomavirus L1 from a rhesus papillomavirus, and they show that production of the protein is below the limit of detection, and if you recode it, you can now get detectable levels of expression. So this is, again, from an expression cassette, I presume using a CMV promoter. You don't get much expression unless you recode it. So these guys probably need a transactivator of some sort to be produced, right? I guess for SV40 it would be T antigen. Is that a fair guess?
Maybe. Yeah, maybe.
And it's always a fair guess for SV40.
And then for the papilloma, there's an analogous antigen as well. So that's basically it. That's pretty cool. Now I remember why Goff sent this to me, because it was right after our discussion of the recoding of the RNA virus. And we said, has anybody recoded a DNA virus?
Right. And these are, with the exception of the retrovirus, these are all DNA viruses, because they typically do this scheduled expression of genes.
Yeah.
Right.
So they use the word fascinating twice.
I love this sentence in the discussion. What exactly is being recognized to achieve the codon usage-dependent transinduction is a fascinating question.
So I labeled this the how paragraph.
We have no effing idea how this works. We're just reporting the facts.
So, Rich, they don't agree with they think that the structure of the RNA has changed, and this messes up the interaction with REV or ORF57. But I like your idea as well.
Yes.
Well, I don't know anything about what's known about how ORF57 works, but REV is an RNA binding protein, right? And has all sorts of other activities that have to do with RNA metabolism and RNA transport and that kind of stuff. And in fact, part of this is that you need a what's the Rev binding sequence also in the recoded RNA in order to get Rev to work.
Right.
So Rev is definitely part of its action has to do with interacting with the RNA. So I see how you could be drawn to an explanation that had to do with either transcription or RNA structure or something like that.
They say codon usage may not be the critical factor, but rather distinct negative regulatory elements imparted by the nature of the skewed codon usage. Well, I think both are possible.
What blows my mind is that it is as if there's a common theme here of the reprogramming the codon usage and using a transactivator to optimally produce the protein at the appropriate time. And yet the codon profile is completely different. So that implies to me that this is like a convergent evolution or something like that. It's a concept that has, maybe there's some ancestral concept and there's divergence from that into different codon spaces. I don't know, but it's weird to me that this same mechanism would be responding to codon profiles that are completely different. And the transactivators that probably don't, well, I don't know. Are the transactivators going to work exactly the same way or in a similar fashion? I don't know.
I think
I don't think so.
To me, the diversity of the viruses involved here, and the fact that there's no cross compatibility between them, suggests that it's convergent evolution. They just all hit on the same solution.
Right. And you just think about transactivators. E1A is not a DNA binding protein. Other transactivators are. So, yeah. The second last paragraph I labeled the why paragraph.
Yeah.
What advantage might this have for persisting viruses? So they speculate on that a little bit, but
I don't really understand their explanation. They say, perhaps in the face of persisting immune responses, highly restrictive temporal regulation may allow the release of at least some progeny virions before immune-mediated destruction.
That didn't really make sense to me either. What makes sense to me is that, if you really want to persist, you want to be able to tightly regulate probably the expression of the final packaging structural proteins, okay? And just occasionally make a little bit of virus enough that it sneaks under the immune surveillance radar or something like that. So this sort of regulation of structural proteins might be key to that.
Yep. All right, there you have it. Anything else?
Cool.
Yeah, it's a cool paper.
That is quite novel.
It is.
Yes.
Very interesting.
I couldn't agree more.
Wait.
Uh-oh.
That sounds like Dickson Despommier.
It does. He's fading in from a distance. He's distant Despommier.
Well, wow.
Oh,
There
now
he
he's here.
Wow. Yep, that's it. Okay.
Hello.
I had to turn on your phantom power.
Ah.
I forgot to wear my phantom mask.
Yeah. We just finished all the papers.
Oh, good.
Yeah, I'm sure you'd like that.
I don't have to reveal my ignorance then.
That's okay. I took your role.
Oh, good.
Let's read some email. And you can listen and comment.
I will.
So here's a relevant letter. Dear TWiV team, it's from Dusan, D-U-S-A-N. We would like to make a few comments regarding episode 351 and the follow-ups in which you discussed attenuation of a dengue virus by synthetic attenuated virus engineering. SAVE. As you discussed, SAVE has been successfully employed for attenuation of several RNA viruses, but it has never been tested on large double-stranded DNA viruses. We wanted to know whether SAVE could also be used to attenuate DNA viruses, so we recoded several genes of gallid herpes virus 2, aka Marek's disease virus, an oncogenic herpes virus of chickens. We de-optimized, optimized, and randomized codon pairs of several essential MDV genes. While we were able to propagate viruses with optimized or randomized genes, we failed to grow some of the mutants that contained codon pair de-optimized genes. To confirm, That codon pair deoptimization was responsible for the observed dead phenotype. We generated additional MDV mutants that contained chimeric genes composed of different wild-type and codon pair deoptimized sequences. Interestingly, viruses with chimeric genes grew with characteristics that correlated with the level of codon pair deoptimization of recoded genes. They don't say which genes. Maybe if they did the glycoproteins, that was the problem.
Yeah.
They shouldn't. This was a few weeks old. Maybe now they know that result. On another note, we share the overall conclusion on the issue that vertebrate viruses that contain codon pair deoptimized sequences are not attenuated because they contain increased numbers of codon pairs that are somehow unfit for efficient protein production, but because these viruses indeed contain increased numbers of CpG and to a lower extent UPA dinucleotides in the recoded sequences. The most underrepresented codon pairs in vertebrates contain CpG or UPA dinucleotides at the codon pair boundary. For example, of the 100 most underrepresented codon pairs, 97 codon pairs contain CpG and 3 codon pairs contain UPA at the codon pair boundary. In addition, 94% and 98% of codon pairs that contain TPA and CpG dinucleotides at the codon pair boundary are underrepresented in the human orpheome, respectively. Logically, maximization of codon pairs with the lowest codon pair scores, codon pair deoptimization, must lead to an increase of CpG and, to a lesser degree, also UPA dinucleotides in the recoded sequences.
Vimmer's theory is taking a real drubbing here.
Yeah.
Yeah.
Sincere greetings from your long-time listeners, Dusan Kunek and Klaus Osterreider, Institute for Virology, Freie Universität Berlin, Germany. I think this is unpublished data, isn't it?
Not anymore.
I know.
Well, we're not an official publication, but
Still.
I didn't find this, although I may have missed it. So, what's the conclusion?
The conclusion is that this codon pair deoptimization works on DNA viruses as well as RNA viruses, right?
They weren't able to grow some of these viruses.
Right. So they're really dead.
They couldn't. They couldn't grow mutants with codon pair deoptimized genes. So you can't answer the question: is it less? I would say it's less virulent.
Yes,
a lot less virulent.
Dead viruses tend to be very virulent.
How does the saying go? Dead viruses don't.
Dead puppies are no fun.
Oh, gee.
Depends on who you talk to.
Dead something tell no lies, right? How does that go?
Dead
Dead men tell no lies.
Dead viruses tell no lies.
Dead viruses tell no tales.
And the other take-home message from this is that we have yet another group that thinks it's the dinucleotide frequency that's important here, not the codon thing. Right. So I thought about this as we were doing the paper from DeRocher's lab, and wondering whether, in that case, it was actually codons that were doing it, or whether it was some other issue of sequence, even dinucleotides. But that would strike me as an incredible coincidence, given that you're with the two different proteins, GP160 and the RRV glycoprotein, that you're actually swapping the codon pairs and getting a transactive, not codon pairs, the codon utilization and getting a transactivators to work appropriately. It seems to me that that's much more likely to be a real codon thing.
Mm-hmm. Yeah. All right, thank you, Dusan. Alan, can you take the next one?
Yes. So I'll take the next one and the reply.
Sure.
John writes, Dear TWiVsters, sometime back there was some discussion on your show about the relative merits of the flu mist nasal vaccine versus the flu shot. If I recall correctly,
did you say nasal or naval?
Nasal. That's the nasal vaccine.
Okay.
Versus the flu shot. Did I say naval?
It sounded like it.
No, I nasal vaccine versus the flu shot. If I recall correctly, you all preferred flu mist for the efficacy. The obvious question, which you didn't address, is whether getting both is better than getting only one of them. Unfortunately, here in sunny Hawaii, the flu shot has been available for weeks, but flu mist is nowhere to be found yet. Is it better to take the shot now and then get flu mist later, to get the shot now and not get flu mist, or simply forget about the shot and to wait for flu mist? One wholesaler I spoke to is not shipping flu mist until mid to late September, but I don't have any firm date on when it will be here in Hawaii. And then somebody, I guess Vincent, asked Stacy Schultz-Cherry.
Yep.
who replied, actually, ACIP, that's the Advisory Committee on Immunization Practices, recommends that people only get 1 dose in a season. The exception is kids 6 months to 8 years old, where 2 doses are recommended. Now, there are exceptions. If the listener received the trivalent inactivated vaccine, receiving the quadrivalent live attenuated vaccine would afford protection against circulating influenza B strains. Also, if they plan to travel in the southern hemisphere, they should consider getting revaccinated. Hope that helps.
Her spell checker did a great number on quadrivalent.
Yes, it really did, didn't it? The quadriceps live attenuated, that would be an interesting place to deliver a nasal vaccine.
On the basis of anecdotal evidence, my wife and I both went and got our flu shots last night.
What did you get?
We got the quadrivalent because we were over 65.
Well, the quadrivalent,
the high dose. They call it the high dose. That's
yes. They get the high dose is for over 65.
Yeah.
Most of the vaccines have now gone to quadrivalent,
which is a live attenuated.
Yes, the flu mist.
Flu mist. Yes,
okay.
That's the live attenuated. The people in official positions always refer to it as live attenuated influenza vaccine or LAVE because flu mist is a brand name.
But the injected high dose is not attenuated?
The injected high dose is a killed virus.
Okay.
An inactivated virus.
Okay, fine.
Sorry, killed, inactivated.
That's right.
Yeah, it's okay.
Remember, Caleb is listening.
I just wanted to comment that there is a major problem. This is not Hawaii specific. There's a major supply problem with flu mist this year. They have had some problems producing the quantities that they expected to have, and so a lot of places aren't getting it. My daughter ended up getting the, I think she got the Sanofi shot, just because the pediatrician can't get the flu mist.
Yeah.
Yeah. So addressing the listener's question, when is flu season?
In
flu
Hawaii?
season, right? Well, okay. Flu season for the United States in general starts. It's not really gotten underway very strongly yet. But the usual guideline is approximately September. Back to school time is a good target date.
Yeah. There's a cool season in Hawaii. It's the level of Mexico. It's the latitude of Mexico.
Right.
And that's now. The flu season is now.
Right.
So I think the priority is to get vaccinated.
Yes.
Okay.
Indeed.
If you wait around for flu mist,
you may end up getting the flu first.
You may wind up getting the flu. So
I agree.
So get vaccinated.
Yeah.
I would also say that now I am not a doctor and I don't even play one on TV, but I don't see that there would necessarily be any downside to getting, if you've been vaccinated, getting flu mist on top of that.
Well, it may not take. In theory, the inactivated is supposed to give you respiratory immunity, so the LAIV may not take. It may be worthless.
Right.
There's also some evidence about efficacy for some of the LAIV vaccines not being as high as it should be.
It's gone both ways. Last year's flu mist was, for reasons that have still not been completely worked out, it was a big disappointment. Mm-hmm. So there was an efficacy study that showed that one had unusually low efficacy. However, previous studies have shown that flu mist has in general higher efficacy than any of the shots. So it's like 80% versus the shot, which is 60 in a good year. So there's a lot of back and forth about the efficacy of flu mist, and was last year just not a good batch, or what's going on exactly. But this year it's probably a moot point for most people because you're not going to be able to get flu mist. But that is an active question.
Okay, we have a few more later if we get to them on flu vaccine.
Right.
This time of year. Kathy, can you take Porter's?
Sure. Porter writes, "Hello, TWiVies. My name is Porter, and I am currently an undergraduate at Skidmore College, and I thoroughly enjoyed this week's TWiV. My
go,
question
Quakers, go!
pertains
Kill! Oh, sorry. No, that's Swarthmore, not Skidmore. Sorry.
Okay, and we don't know which week's TWiV he's talking about, but maybe it'll become clear. My question pertains to potential uses of RNAi to treat viral infections after the infection has taken place. This could be useful for viruses that cannot be vaccinated against, or places where the vaccines couldn't reach the hosts before infection occurs. An example of this is hepatitis B, and with this example, there are many ways we could conceivably target the virus using RNAi. For example, its genome and messenger RNA. However, the idea of this brings up many questions, one of which being whether or not the RNAi machinery is actually strong enough to destroy the viral infection at a rate which would allow the immune system to take over. The RNAi machinery would have to overcome all of the RBPs associated with the virus. RBPs...
binding proteins?
RNA binding proteins? Something
Yeah.
like
RNA binding proteins. Thank you.
RNA binding proteins.
So the RNA machinery would have to overcome all of the RNA binding proteins associated with the virus and cleave the RNA to a length that nucleases would be able to degrade it effectively. My main question is whether we can overcome this by targeting multiple sites on the viral RNA at once, thus increasing the rate of cleavage, assuming the machinery does not become saturated, and additionally target endogenous proteins that allow the virus to propagate. Obviously, we cannot target RNA polymerase II, but using the hepatitis B virus example, could we feasibly target the bile acid transporter, which is required to transport the virus into the cell, to a level that is not fatal to the host, and possibly combine that with targeting the messenger RNA and viral genome, thus lowering the viral presence to a level that allows the immune system to take over? I hope that this idea is not too far-fetched. However, I believe the consideration of this is in order. Thank you, Porter.
That's good. So this came in on September 16th, and that episode that week, we talked about feline immunodeficiency virus, and one of the so that was in response to a letter, and the letter writer had asked whether you could use RNAi to treat infected cats.
Right.
So there you go. I think this is a fine idea. I think people are probably doing this, right? Targeting multiple. I'm not sure about receptors because that could have side effects, but you can target multiple virus genes, right?
Sure.
There's no doubt about it. It's just a matter of which ones and doing the trials that prove it. But certainly RNAi therapies are in trials, right?
Yep.
What I like about this letter is that Porter understands all this stuff well enough to understand all the caveats.
Yes.
Okay, you can't just stick an RNAi in. It's got to somehow get past all the stuff that would bind up RNA and degrade it and et cetera. You can't just go knock out a receptor because you might do damage to the host. Maybe you can knock it down a little bit. He understands all the subtleties, and that's great.
Yes.
So here is a question, Porter. I want to see if you're still listening. Where did you learn this? Did you have a great course at Skidmore, or is it from TWiV? Tell us.
Yep. And apologies again for screwing up Skidmore and Swarthmore. I've done that before. Not on this show, but...
Rich, can you take Trudy's?
Sure, Trudy writes, I ran across this paper in Nature and thought you might find it interesting since it is a frequently discussed topic. And this is... once again, about
postdocs.
About postdocs. It's about the postdocalypse.
Postdocalypse?
Yeah. And in this particular case, the issue is postdoc salary and in particular benefits. And I think one of the issues here that's made in this article is that when the postdoc was invented, it was conceived of as a temporary training opportunity past your PhD to prepare you for your life in research. When I was considering postdocs, I didn't think of a postdoc as a job. Okay? It was more training. It wasn't a job. And that's how they were compensated. You got a modest stipend and virtually no benefits. And what's happened is that the postdoc as an employment opportunity has evolved, but the compensation has not.
and even at the time you were going through it, there's some evidence that the that it was really more of a stopgap measure to handle an initial oversupply of PhDs, even at that time.
Oh, that's interesting.
Because if you go back another generation, it is very unusual to see anybody whose resume includes a postdoctoral position. So people would come out of a PhD program and they were presumed to be ready. They were presumed to be ready to initiate their own research. And in fact, PhD programs in general have retained the same level of training. So this notion that somehow after graduate school you weren't quite ready was, I think, fabricated to cover a fundamental problem in the employment structure.
You know, there's a good box here about what is a postdoc, and they say in some universities they are employees and they get standard benefits, but in others they are not. So there's uneven classification of what exactly they are.
So in medical school, when you graduate, you become a resident, which is equivalent to a postdoc position, and they routinely were so underpaid and overworked that they formed a union and they got rights, and the next thing you know, it's a nice paying job now, and it's not such a bad thing to be an intern. The same is true.
I wouldn't say nice paying job exactly. It's still an indentured servitude, but there are rules about what you can and can't do to them, which was not always the case.
So my experience was totally different than Rich's because I took something called a guest investigator position. At Rockefeller, they didn't call them postdocs. They call them guest investigators, meaning like guest workers. You come in, you do your work, now go home and don't stay here. I took my postdoctoral position with the thought in mind that they could find me a job after I finished showing them how great I was. You can't do that in your doctoral work. I mean, sometimes you can, right. but mostly it's the connection between your major professor and the next level of your life that can be the connector. And that I think that was a stepping stone. I always looked at it as a stepping stone. I never heard people complaining about it until, maybe 10, 15, 20, 15 years ago, maybe, when people began to make permanent jobs out of them. And permanent jobs for postdocs are just not applicable. That's just not appropriate. That's a person like the man who came for dinner and just never left. That's basically the way these positions are perceived, and that's probably incorrect. So maybe there should be some position where you can make a living. The incentives aren't great to stay in that job because the pay isn't that great, but it's a living wage, as they would put it. And you're certainly more skilled than somebody who just came in off the street. So,
well, you don't have to constrain it by wage. You can just say this is a 3 or 4 year position and pay appropriately.
Yeah,
I don't think, because as this article points out, there are many people doing postdocs. They have families and they have to take care of them. I know. And it's not fair to keep them working for $30,000 a year.
But the fundamental problem here is still that training and staffing are linked.
Of course.
And until we separate those two things, until we say we're going to staff labs with full-time employees and not pretend that they're actually training, then we're going to continue to have this problem, because you're going to keep cranking out people who are trained for jobs that don't exist.
But you want to not have postdocs at all? Is that what you're saying?
Ultimately, I'd like to eliminate the postdoctoral position entirely, produce a whole lot fewer PhDs.
Right.
So doing a PhD would be an unusual thing to do. And what you'd do if you wanted to work in a research lab is you'd come out of your undergraduate program and you'd get a job on some kind of a technician career track and move up through the technician career track. And if you decided that you wanted to be a PI, then you might choose to go to graduate school, but it would not be the standard thing to do if you wanted to do research. And until we do that, we're going to continue to have this problem where we just have way too many people who have not to be too melodramatic, but essentially sacrificed the best years of their lives to train for a job that doesn't exist.
On a slightly different tangent from this, just thinking about the fact that postdocs are neither students nor employees. When I was a postdoc at UCLA, we had the lamest of ID cards. It was this piece of paper that was typed. Now, years before that, I'd been an undergraduate and had a plastic ID card at Purdue. So having this piece of paper was really amazing. And one of my friends was stopped by the UCLA police for jaywalking. And they asked him what was he doing on campus, and he had to pull out this stupid
piece of
paper ID card
that
really did not, do well. But I mean, that's just a sign of, there's some good things and bad things about it. You don't have to go off to faculty meetings. You don't have to go to classes. You can spend all your time doing research. But then you have this status that is neither faculty nor student, and
we got no ID whatsoever.
This has been pointed out in other discussions and correspondence that we've had, is that the whole concept of a postdoc is treated quite differently by different institutions. And one of the things that might be done to address this problem is for the whole notion of a postdoc, or for postdocs to organize themselves and institutions to organize offices of postdoctoral research or whatever in a much more uniform fashion, to give this whole thing
I think that's the order in which it will have to happen. The postdocs will have to organize, meaning unionize, and threaten the university in order to form these offices to manage the program.
Some universities have very good postdoc offices and programs and stuff, but others have nothing.
And aren't the postdocs in the University of California system now unionized?
There are some schools that have unionized, where the postdocs have unionized.
Anyway, check the article out. I don't know. Is it open, Alan? Can you tell?
This one, yes. This one I did not log in.
All right, very good. Next email's from Anthony, who wrote a letter to the International Federation of Pharmaceutical Manufacturers and Associations, asking them, did the recent avian influenza outbreak in the U.S. Midwest in any way hinder production of egg-based vaccines? And he writes, they did not respond to my email.
Don't feel bad. They don't answer mine either.
But then he sends a link to a news article. He says, this claims that the Midwest avian influenza outbreak did not affect vaccine production. This is an article in the Seattle Times entitled, Egg Shortage Won't Affect Supply of Flu Vaccine. But maybe it's affecting flu mist. Is that possible?
No.
That's some other problem they're having.
Right. That's
the virus, the strains aren't growing well.
I think it's that the strains aren't growing well in the case of flu mist, which is, I don't think, produced in eggs.
It is.
It is.
Yeah. Oh, it is. Okay, that's right. But it's a separate issue with that. The thing to remember now is flu production season is done.
Yep.
These companies finished producing this season's vaccine in July and August and shipped it. So with the exception of the flu mist, which is still trickling out the door.
Flu mist made by MedImmune. We have a listener at MedImmune. Ken, if you can, let us know what's going on. Yeah, I'm sure he'll tell us.
My very first graduate student is a director of biological quality control at MedImmune, and he is constantly running around checking the egg supply and figuring all this stuff out. I was tempted to write him about it. Well, I will. I'll write him about it, see if I can get an update on what's going on with flu mist.
Alan, can you take the next one?
Yes, Jim writes, Margot and I just got our seasonal flu shots. She got the senior version with 2 strains, but this is when we first heard about the quad vaccine, which seemed like a better idea and is what I got. I see online there's also Flu Block that is 3 strains but 2 types, the A and B versions. The nasal spray is 4 strains. Is this evolution of flu vaccines part of the personalization of medicine? It's starting to be confusing. Seems to me there should be a senior version of the quad vaccine like the trivalent vaccine. Will we see a patch option next that's available in several versions? We've seen lots of signs urging us to get flu shots, but nothing that sorts out the options and reasons for selecting one over the other. And Jim is our friend in Smithfield. Well, Jim, it used to actually be more confusing because the recommendation was split among different groups. So people over 65 were recommended to get a flu shot, and people under 65, but who were not kids, could get a flu shot, but it wasn't specifically recommended, and yada, yada, yada. And the 2009 swine flu epidemic changed all that, and we went to what's called a universal recommendation, which I think was the standard in Europe for years before that, which is everybody over the age of 6 months should get a flu shot. So in that respect, it's gotten simpler, but you are correct that there's a diversity of options there. I guess we're going to have flu shot Grove Stand coming out next. Diet flu shot.
Organic,
gluten-free.
Flu shot.
Vegan.
Yes, gluten-free, vegan.
Non-genetic. I mean, genetically modified. No. What would they say about that?
The A and B influenza viruses, the traditional vaccine would contain 2 influenza A strains and a B strain.
Right.
The quadrivalent contains 2 influenza A strains and the two circulating B strains.
Mm-hmm.
So with B, you either get one or the other, essentially, circulating in a given year. So by adding the quadrivalent, we could essentially, vaccinate against any conceivable influenza B virus that would come along. The A is more diverse, and so you get 2 strains of that in there. I don't know about any kind of a bivalent vaccine.
Me neither.
With only 2 strains. So I think the high dose. Now, what all the manufacturers are moving toward now is quadrivalent. So there will be 2A and 2B strains, ultimately in all versions of the vaccine. So these trivalent vaccines that you're seeing are just going to stop existing, and there will only be quadrivalent vaccines. It's a better approach.
How many companies make them?
About a half dozen.
Yeah.
The elephant, the 800-pound gorilla, I guess, would be Sanofi.
Okay.
Sanofi Pasteur. They make
at the station that we went to last night at a CVS pharmacy. Just gave us 2 choices. One was under 65 and over 65, and the over 65 was called the heavy, and so we took it.
Yeah.
Do you know what the manufacturer was? Who the manufacturer was?
I didn't notice.
So the Sanofi, I think it's the Sanofi product, is Fluzone.
Mm-hmm.
Yes, the most likely thing that you'll get if you go anywhere was what my town's free clinic gave out. I'm pretty sure it's what my daughter got at the pediatrician. I think it's what my wife got at CVS. Is the Fluzone. That's the big product in the market. But then there are a bunch of other inactivated vaccines from several other companies that have smaller shares of the market. And then there's FluMist, which of course is the live attenuated vaccine.
So, Fluarix quadrivalent is made by GlaxoSmithKline. Right. Flulaval quadrivalent is made by ID Biomedical of Quebec. Fluzone variants are made by Sanofi. Afluria is made by BioCSL. I think that's Australia.
Yep.
Fluvirin is Novartis Vaccines and Diagnostics. More Fluzone. Flucelvax is a cell-based vaccine made by Novartis. And Flublock, the HA made in insect cells, made by Protein Sciences. Those are the companies, and MedImmune. But CDC doesn't provide a lot of guidance, that's for sure.
So how's Flublock manufactured?
It's produced in baculoviruses, in insect cells.
Okay.
HA, production of the HA, which makes virus-like particles.
Oh, interesting.
And Alan made a joke about, sugar-free flu vaccine. I have a bar on my desk here. It's got on its label gluten-free, dairy-free, soy-free, vegan, and kosher. Is there anything they're missing?
So when you open it, there's nothing inside.
It's empty.
It's non-GMO, right?
No, it's got to be non-GMO.
Well, we talked about the non-GMO salt, right?
Yes, right.
Right.
Yes.
That's
Doesn't
right.
say non-GMO.
I wouldn't touch that with a 10-foot influenza virus.
It's called Larabar.
Are they?
I like it. I'm eating it because my kids wouldn't eat it. We bought it for them. So I'm the garbage disposal.
So you're the Mikey of your family.
Let's see. We'll do 2 more. The next one is Kathy, which would be Joaquim.
Okay. Hi, Twix. Thank you for your great shows. And I'm glad to hear that you, Vincent, like my tip about Dan Carlin's podcast. Here in Stockholm, Sweden, fall has almost just begun with mean temperatures around 12 C, however, still with quite fair weather. I think that it needs to be taken up that also young scientists can do awesome things. This year's grand prize in the Google Science Fair goes to Olivia Hallisey, 16 years old, for her Ebola detection system. Pretty cool, huh? I'm not informed enough to understand whether this is a new invention. However, it still tells the tale of young people being interested in science and in doing good. Just to compare with the last week price bumps in TBC, which I think is the TB cycloserine drug, tuberculosis drug, and toxoplasmosis drugs, which might put some shade on the drug industry. Right. And so this ended up being the listener pick last week.
Right.
And the it's a really cool device. And as far as I know, it's probably a new invention. Just putting together these, you just add water basically, and you can figure out whether the person has Ebola or not.
Yeah, it's a paper-based detection system where they're looking for antibodies, and she's put an antigen on the paper, and she stabilized it with silk.
Yeah.
Right, which we've talked about for stabilizing vaccines before. That's being tested. And so she used it to do the silk. It's very cool. So, and it's sort of like an ELISA, basically, a paper-based or a solid phase-based ELISA, where, you add things around on the paper and they float towards the center. But the key here is that she stabilized it so it could be, could sit out in the heat and still work. It's great. I think it's great that a 16-year-old would do something like this.
Yeah.
And her trophy looks like a Lego, doesn't it, Kathy?
Yes, that's exactly what I think it is.
It's very cool.
Really
it's
cool.
great. Yes, that was a pick last week. I forgot to see this. All right. Congratulations. One more. Rich Condit.
Jessica writes, Dear TWiVologists, I began listening not too long ago and have thoroughly enjoyed your show. I'm not sure how many of your listeners are in risk management careers, as I am, but when you mentioned that particular perspective for studies of biohazardous agent releases during building collapse, I thought I'd let you know that I am a listener from that community. More specifically, environment, health, and safety, and even more specifically, biosafety. Unfortunately, I have not heard of or found many studies of this type. But I did find a wish list of studies from the Department of Homeland Security, which basically gives some resources for who to turn to in such an event, general testing recommendations, standards, etc. Maybe this document will give some listeners in research ideas for grant proposals. And she references a document from the Department of Homeland Security on basically how to deal with hazards in peculiar circumstances. It's a study group, some large committee looking at this problem. As someone who is involved in conducting risk assessments for research, much of which involves viruses, I'm always picking out your virologist opinions on the risk profiles of viruses you talk about on the show. As a promoter and practitioner of biosafety, I think the show could use the opinion of a biosafety expert from time to time. In fact, there have been many recommendations to include an MD on the show, and if this is a possibility, perhaps consider an MD with expertise in occupational health, particularly around exposure to viral vectors used in research or clinical trials. As a member of the American Biological Safety Association, I belong to their listserv, as there's often useful or interesting chatter there. It's free, and you don't need to be a member to join. Maybe a good location for picks, etc. I picked an article that came up today that combines the recent discussion topics of transmissible cancer cells and risk management. I thought it might generate some interesting commentary from the panel, particularly because there have been cases in which exposure to human cancer cells generated tumors in immunocompetent individuals. I'll leave it at that, and she provides a link to this article. I confess.
Yeah, I couldn't get in.
Paywalled.
I couldn't either. Yeah.
I'll leave it at that, although I'd like to reference a question I'm still looking to answer from a previous email in regards to mycoplasma. Why isn't this being engineered as a potential tool for immunotherapies, etc.? Ideas? It's currently 13C, 80% humidity, 14 kilometers per hour, and as Neil deGrasse Tyson told us last night, we're on the verge of a rare harvest, blood moon and lunar eclipse tomorrow night. So this is from a while ago. Right. He actually quite enjoyed, well, he was actually quite annoyed at the proclamation that this was a rare event. So, thank you for your awesome insight. P.S. Thank you, Dr. Spindler, for representing lady scientists on the show. We need more super cool, intelligent scientist role models who are females represented in our culture for many reasons, and I love that you are a panelist. Sincerely, that was Jessica. Is that right, Jessica?
Yeah. Well, I couldn't get to that one cancer cell thing.
No.
The mycoplasma question.
Nobody wants it in their lab.
We should. Yeah, I don't. Well, I'll ask that on
Or their
TWiV.
lungs, for that matter.
Don't know. Did you ask that before, Jessica? I guess so. I have no answers, but I will bring it on to TWiV. How's that?
There you go.
Let's.
The blood
yes.
moon lunar eclipse was really cool.
It was.
Yeah, you sent us a picture, right?
That was my pick.
That was your pick?
That was my pick.
Okay. All right. Kathy has a hard out, so let's let her do her pick first.
Okay, so I think everybody's picked something about the Martian already. I
I
can't
have
remember,
not
Alan,
yet.
whether
I'm
you
still,
had.
I'm still waiting to pick something about the
Okay.
Martian, I guess.
So I made my favorite Martian pick. First of all, my favorite Martian was one of my favorite TV shows when I was a kid. So my pick is it's about an hour-long video, but it's really fun to hear Andy Weir, who's the author, with Adam Savage, and Mark Hadfield, who's our Canadian astronaut, talk about the Martian. And so you get a little bit of the real astronaut's view, and would he have done the same thing as the I can't think of her name, the captain or whatever she was of the Mars ship that had left Watney behind. Would he have done the same thing? And he talked all about the decision making and how when you're in space, or even at the International Space Station, he relates how somebody says, Earth says we need to do this. And so there's this dichotomy between Earth and the space station, and that it's going to be even more so with Mars, and so how that affects decision making and so forth. Anyway, it's just a cool interview.
Neat.
I actually watched the whole thing, Kathy, and I thoroughly enjoyed it. And I once again find myself impressed with is it Mark Hadfield or Chris Hadfield?
The astronaut.
Yeah,
Mark Hadfield's a politician, I think.
Oh, yeah.
I think
it's Chris.
It's Chris.
It's Chris. Yeah.
Chris.
Yeah.
Yeah.
The link was goofed up then. Yeah.
So I once again find myself quite impressed with him. You know, he's out there. He makes sure you know he's there. But I think he's doing a real service for space science. And he's really very thoughtful and very articulate. And it's really fun to get his insights into space travel from the point of view of a career astronaut. It's great.
Did you read his biography? Did somebody pick that?
No.
I read his biography. So, yeah.
So, you know, we came up with this expression, I don't know how long ago, but the expression could actually come true. That is, when two opinions differ by a wide margin, they could be worlds apart.
Yes.
That's weird.
Kathy, do you want to go now?
I'll keep listening for a little bit. That's all right.
All right, Alan, what do you have?
I have another space-related thing. So there's this Project Apollo, which has taken all of the photos from the Apollo missions and scanned them and put them online.
Nice.
Now, as you know, the astronauts took cameras, and like any good tourists, they took lots of pictures. But NASA only released, they obviously, you've got amateurs shooting photos inside a confined vehicle for the most part, and a lot of them didn't come out very well. But
even
NASA,
with a Hasselblad?
NASA has traditionally released, the Earthrise and the other good stuff. This is a complete collection, and they're just organized by which film magazine they're in. So Apollo 7, Magazine 3, Magazine 4, and so on. And it just goes through, and you can go through each roll of film.
Boy, they didn't edit any of these.
Nothing is edited, so it's a lot of, finger in front of the lens, reflection, all the outtakes.
Cool.
But then there's also a lot of stuff that's cool that just never, saw the light of day because it wasn't relevant to whatever they were doing at the time. And this is now digitized and online. And as a result, people are already remixing it into stuff because, of course, this is government product, so it's all public domain. And someone has already made an awesome YouTube video of some photos from these missions and set it to a little bit of background music and mission audio, and that's just an example.
Cool. Very neat. Beautiful photos.
Yes.
Very nice light. Rich, what do you have?
I have something that I ran across a while ago. It's a feather in a bowling ball dropping vacuum. Okay, so this is Galileo's old experiment where he said that objects of different masses will fall at the same rate, and the extreme of that is to look at something very light like a feather falling next to something very heavy like a bowling ball. But if you're going to do that experiment, you need to do it in a vacuum. As I recall, we had an evacuated tube with a ball bearing and a feather in it at one point in science class to demonstrate some of this principle. But to do this, what's entertaining about this, the experiment is done in a facility called a space simulation chamber at the Space Power Facility in Ohio. And this is a giant, I forget how big, but it's big enough to hold, a small spaceship. A giant chamber that can be evacuated. It's the largest vacuum chamber in the known universe. And this was developed in order to look at the effects of the vacuum of space on, I suppose, machines and et cetera, and to test space vehicles and et cetera. I don't know how much it gets used now. These guys look pretty excited to do this experiment in it. And what they did was to haul a bowling ball and a feather up to the ceiling of this thing and evacuate the chamber and then let them go. And sure enough, they fall at the same rate. And it's a cool
They'll never be able to get it published in a prestigious journal, though, because nature abhors a vacuum.
Oh,
nice.
Yeah, they do it with air first.
Yes.
And then they evacuate it.
So they do the control.
They did this
very
on the moon.
effective. I just love
Very
this
good.
video.
They did it on the moon also.
And I like the little bit at the end where he points out that Einstein had a different perspective on this experiment, which is that the reason they seem to fall at the same rate is that they aren't actually falling.
Okay. Yeah, it's very cool. Nicely done. Dickson, what do you have?
Well, I have a website from the state of California talking about the drought and all the things that they're doing to ameliorate the drought. And it's in its 5th year, and it doesn't look like it's getting any better. That is to say...
The drought or the website?
Not either one, actually. It's not a very great website, but it's got a lot of topics there that were not on that website 3 or 4 years ago. And one of them is reusing wastewater that humans create, and that was an abhorrent possibility. You couldn't even mention it 10 years ago, and people would just, turn their backs on you. And now they're embracing this as a new technology. It's not so new, but the state of California is going to set a lot of trends as to how to deal with droughts because they want to, and they have to.
Is there going to be a Niño thing that
El Niño?
This? Yeah.
Well, if there is, it's not going to take care of it. It's going to flood everything and wash away all the topsoil. So that's number 2, strike 2.
How about they stop using open air irrigation?
Oh, that forget your almonds then. If you don't like almonds, that's good. But if you do,
but you could irrigate a lot more efficiently. So.
Oh yeah, absolutely right.
On this website, Dickson, in the banner at the top, they show contrasting photographs at different times of a couple of different reservoirs and stuff.
Yes.
And during the last couple of years, I've driven a couple of times Interstate 5.
Right.
Through Northern California and Oregon it goes through the reservoir that's associated Lake Shasta.
Yes.
Okay. Yes. And it's just so dramatic.
That's right.
How low that lake is.
Exactly.
You can really see it. You can see it in these banner pictures. It's a different reservoir, but it's nearby the Lake Shasta.
Things got so bad that they had a fish hatchery, I forget the town, but they had to transport the fish to a lake as an emergency measure just to keep them alive so they can bring them back whenever the water comes back.
Which could be in 500 years.
Exactly. Yeah, it's sad. It's very sad.
My pick is a blog post by John Eisen, who's a professor at UC Davis. It is entitled, Does AAAS Care About Ebola Anymore? Nope, and probably never did. And it's just, he's a big open access proponent. He doesn't publish in a journal that is not open. And he says, back in October, he criticized AAAS and ASM for trying to give themselves a pat on the back for making a few papers about Ebola freely available. So he decided to check and see what was going on, and he found a bunch of papers are all behind paywalls. So much for helping the world, he concludes. So, I think it was a publicity stunt to a certain extent. They've stopped doing it. They're closing it, It's too bad.
And by the way, I realized after a recent episode that we should have called out the New England Journal of Medicine, which I don't know if this is a uniform policy, but I think every Ebola paper that we've talked about from them on TWiV has been open access. Mm-hmm. So they've actually, and they're in the same position as AAAS in terms of making their money from subscriptions, but they've, I think, done the right thing. Now, I'm sure John Eisen will correct me if I'm wrong, but my experience of them, the Ebola papers that I've wanted to get from them, have all been open.
So, if you go to the WHO website, can you find these paywall articles for free on their site? No matter what, they're gone.
No, you're not allowed to do that, Dickson.
Sure you are.
No, you're not.
I would do it.
The journal has to open it up.
Well, they may have done that, but then no one would have known about it anyway, right?
All right. We have a couple of listener picks, and this week we have 3 compared to a few weeks ago when we had none. Thank you. Anonymous sender sent a New York Times article entitled 732,000 American Lives Saved by Vaccination, according to a recent estimate by the CDC. Mm-hmm. And from Paul, from the Saturday Morning Breakfast cartoon, he sends a cartoon, which is lovely. Science Articles, a guide. And you should read it because I cannot. This is a family show.
It's pretty good.
It is good.
Very funny. And finally, Judy writes, Dear Virus Virtuosos, thought you'd enjoy this. Maybe a listener pick. And this is an animation from openculture.com. How a virus invades your body. It's a previously made video with narrated by NPR's Robert Krulwich. I've seen this before. It's kind of nice. It's got a few errors, but we won't worry about that.
Yeah.
Did anybody see the new series on Nate Geo on Breakthrough?
Nope.
No.
Because the first episode was on the Ebola outbreak, and it was very, very well done.
Oh.
And
extremely well done.
Judy's, as usual, thanks for all you do, and thanks for keeping my retired brain working. Judy, retired high school teacher in San Diego. Today, rain. Slight winds, 7 miles per hour.
Rain in San Diego?
Perfect room temp. Maybe when I was there, it rained.
Gee.
Mm-hmm. All right, that's
odd.
TWiV 362. It's at twiv.tv and iTunes, and do send us your questions and comments. TWiV at twiv.tv. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Have a good trip.
Thanks. I will.
Dickson Despommier, he's here at Columbia University and verticalfarm.com. Thank you, Dickson.
Yep. Sorry I was late.
No problem. I enjoyed it.
Oh, good. Me too.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure enough. Always a good time.
And Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.